The next speaker is Todd Macfarlan. He is a professor at the NIH, and Todd has a very strong background in gene regulation, so he's going to expand on that theme now, moving kind of downstream from Sonic Hedgehog.
Thanks, Sam. So my laboratory is really interested in transcription factors, and this is actually just a modern take on Waddington's epigenetic landscape, in which you see a ball rolling down the hill and generating different types of cells. So we're really interested in what are the factors that actually drive the end stage of this, which is forcing cells to go down into these valleys to choose their actual phenotypes. So this is just a plot of the main 36 or so mammalian transcription factor families. And basically in the top, we just counted the number of genes within each transcription factor family. And then on the bottom is actually a ranking. This was actually done from Kyle Siebenthal and John Stam's lab, ranking transcription factor families according to their tissue specificity. So factors on their left here are those that are the most tissue specific, meaning that they're expressed in the least amount of different tissues in human fetuses, to those that are the least selective, in other words, factors that are expressed in most tissues. And the thinking is that these are transcription factors that are involved in important homeostatic processes, whereas the transcription factors on the left side are those that are important in driving cellular differentiation. So today I'm going to talk about two of these families, the homeodomain family and the helix-loop-helix domain family, which are involved in driving motor neuron differentiation. So I'm going to just summarize in the next few slides the classical experiments that basically showed that transcription factors are essential for driving cell fate. So first, from decades worth of work, many groups have shown that transcription factors mark developmental stages. In other words, if you generate antibodies or use in situ hybridization probes, you find that transcription factors will mark distinct lineages during embryo development. And I'm just showing here the classic pattern of the differentiation of ectoderm from embryonic stem cells, which then go on to generate neural stem cells, and eventually these will differentiate into motor neurons. And you can see that there are distinct transcription factors that actually mark these various stages. But just because they're expressed in different times in development doesn't mean that transcription factors are essential. It really comes from genetics experiments showing that if you knock out transcription factors, you lead to these phenotypic consequences. And a good example of this is Sam, when he was a postdoc, knocked out the islet-1 transcription factor and shows that in these mice you don't get motor neurons. So this shows that not only does a factor like islet-1 mark the development, it also is essential for driving the phenotype. Now, an intriguing feature of mammalian development is that these same transcription factors often get reutilized in distinct developmental contexts. So in other words, a factor like HB9, which we use as a marker of the motor neuron lineage, is also expressed during endoderm differentiation. So how can this same transcription factor, which has the same biochemistry, lead to these dramatically different outputs in changing gene expression patterns? So we're really interested in understanding the sort of fundamental principles and how transcription factors work and can control such diversity. Now, I'll also point out some very important classic experiments that just demonstrated that transcription factors can also reprogram cell fates. And this is actually something that we're interested in the laboratory. So there was the classic experiment of taking a fibroblast cell and expressing the transcription factor MyoD and showing that this could convert the cell into a muscle. So a single cDNA expressed in fibroblasts can dramatically change the phenotype of the cell. And then around 10 years ago or so, the Yamanaka experiment demonstrating that you can also take a fibroblast and put in a bunch of transcription factors like OCT4, SOX2, KLF4, and MYC, and you can actually reprogram fibroblasts back into a pluripotent stem cell. And obviously, this has been a revolution in regenerative medicine. Now, we also know today, which I'll talk a little bit about, is that you can also take pluripotent cells like embryonic stem cells, force the expression of transcription factors, and generate the end cell type without having to go through all of these intermediate stages. So the questions that really interested me in my laboratory then are, how do such a limited set of transcription factors achieve such diversity in outputs? And the sort of corollary to this, which is appropriate for this meeting, is to think about this in terms of regenerative medicine, is can we use transcription factors to better engineer cell types with improved efficiency? So that's going to be the sort of heart of what I hope to achieve in my lab over the next 10 years or so. The problem, however, though, is that we really need a better understanding of what are the determinants of transcription factor binding. And I took this from a nice review from Slattery and Tibbs last year, in which we've been able to basically categorize transcription factors according to the DNA sequences they like to bind. And this comes from various types of in vitro assays, like classic EMSA assays, and other types of in vitro binding data. So we basically have these libraries of transcription factors and what are the DNA sequences they like to bind to. And then what we can do is we can predict, well, where in the genome do these sites actually show up? And then we can make all these predictions about where a transcription factor might bind. And you can see here, for example, this is going across a chromosome. Each of these represents a potential peak, for example, in a ChIP-seq data set. But in reality, we find that transcription factors often only bind to a very small subset of those sites that are actually potentially, that have the potential to be occupied. And in some cases, a really good example is the Hox genes, for example. They have a preferred binding site that they actually never associate with in vivo. So that basically tells you that we don't have the right tools to be able to predict how transcription factors will interact with DNA. And part of the reason for this is that we also sometimes fail to forget that DNA is itself a three-dimensional structure. It has its own structure, and here we actually show nucleosomes packaging the DNA. And in blue, for example, we're highlighting those potential sites that could be occupied by a transcription factor dimer. And you can see that in this model that what we were interested in knowing is why does a dimer like this particular one bind to some sites and not others? And it really requires us to have a lot more understanding of how transcription factors are affected by things like the chromatin structure and nucleosome architecture. So the model system that we're using to try to address how transcription factors interact with DNA in a chromatin context is to use motor neuron development as a model. And there's several reasons that this is a good model. First, as you heard about in the last slide, we know quite a bit about the signaling pathways that are involved in the differentiation of motor neurons. Two of these signaling pathways are the sonic hedgehog pathway, but also signaling from the axial mesoderm, in this case, retinoic acid signaling. The second reason the motor neuron model is nice is that you can actually differentiate embryonic stem cells into motor neurons from the mouse in about 6 days, and it basically largely recapitulates the signaling that's happening during normal development. So you can take ES cells, you can grow them as embryoid bodies for a few days in the absence of LIF, which is the factor used to keep cells pluripotent, and then you can add in retinoic acid and sonic hedgehog or smooth antagonists, and you can generate about 30% of motor neurons in about 6 days. The nice thing about this model is that you can basically generate millions or even hundreds of millions of cells in a week, whereas in a typical embryo, you get about 3,000 motor neurons. So this is nowhere near enough to be able to do biochemistry kinds of experiments, but at least in vitro with this model, these experiments become largely within our grasp. Now, the other nice things about the motor neuron model, which makes it exceptional for studying, is that we know a bit about how some of the transcription factors actually work to control motor neuron differentiation. So the factors that our laboratory has been interested in are these limb homeodomain transcription factors islet1 and LHX3, which are downstream of these signaling pathways, and they bind to a motor neuron enhancer found at the HB9 gene. These transcription factors make a hexamer complex, which synergizes with adjacent transcription factors. Here I'm showing an example of the BHLH transcription factors neurogenin-2 and E47, which bind to E-boxes, and all of these factors in turn synergize to activate expression of the HB9 gene. Now, this has largely been done through experiments with electroporated into chick embryos, but we know very little about how these factors actually associate with DNA genome-wide, and that's the question that my lab is interested in addressing. Now, I should also point out that work from Sam's lab, primarily from Soo-Kyun Lee, demonstrated that if you overexpress these proteins, neurogenin-2, LHX3, and islet1, into a chick neural tube, you can basically force the ectopic expression of motor neurons. So this is kind of like the MyoD experiment that shows if you just force the expression of the right factors, you can drive a particular phenotype in development. So we're really interested in trying to figure out what is it that these transcription factors do, how do they synergize with each other genome-wide. So this is the setup that we're actually using in the laboratory. The first is to take an embryonic stem cell line and to force the expression of single or combinations of transcription factors and look at how does the addition of other factors affect the binding of a factor in study. So for example, if we overexpress islet1, or if we overexpress islet1 with LHX3, how does the appearance of the second factor affect the ability of islet1 to interact with its target DNA? And then you can see you can obviously build up on this by adding in additional factors. The second set of experiments that we're attempting is to see how does the same panel of transcription factors interact with DNA if you start from a distinct epigenetic state. So in other words, if you start from an embryonic stem cell and you add neurogenin 2, islet 1, and LHX3, we expect that this is going to turn on motor neuron genes. But what happens if we turn on these same 3 factors from a different kind of cell, which may be less plastic? So we need a good system to be able to reliably induce transcription factors. So the way we do this is by using this elegant system that was adapted by Michael Kaiba's lab at the University of Minnesota, in which they recreated this modified locus at the HPRT region on the X chromosome. And the gist of this experiment is that it's an inducible cassette exchange in which you can clone your gene or gene of interest onto a plasmid. You can then transfect these into embryonic stem cells, and you basically swap out what's currently at the locus, in this case a Cre recombinase, for your gene or gene of interests. So you can put in a single gene, multiple genes into the same locus in single copy in an embryonic stem cell line. And the result then is a cell line in which you can add a drug to induce your gene or genes of interest. So in this case, we're using a doxycycline system that binds to this RTTA, and then you can lead to activation of your target transgene. So what we've been putting into this locus is actually combinations of transcription factors. And this is just an example of one of the cassettes that we've built. This is a polycistronic cassette that expresses 3 transcription factors. In this case, neurogenin 2 with a V5 epitope tag, islet 1 with a flag tag, and LHX3 with an HA tag. Now, these tags are really nice because we can basically track these proteins, and eventually we can use the antibodies against these proteins to map how they bind with DNA genome-wide. So we've just really begun these sets of experiments, and I'm going to show you some of the preliminary data. So first, what happens when we induce individual or combinations of these transcription factors in embryonic stem cells? Well, first, this is just to show you that the system actually works really nicely. If you just have an islet 1 cassette and you add doxycycline, you can see nice induction of the islet 1 gene. Likewise, if you put in islet 1 and LHX3, you can turn these guys on. And further, if you add neurogenin 2, you can turn all 3 genes on at once. So what happens to the cells 48 hours after you turn on these distinct panels of transcription factors? Well, the bottom line is that if we turn on islet 1, we're basically turning off and on a whole set of genes. But we were actually surprised to find that when you turned on islet 1 and LHX3 in combination, which has been shown to be able to drive high levels of HB9 in a developing neural tube, these had very little effect, actually, in embryonic stem cells. Likewise, if you added neurogenin 2 on top of this, we were expecting to see more synergy and that you would get activation of a lot of motor neuron genes. And in fact, we see very little changes again under these conditions. But if we add retinoic acid on top of the NIL cassette, you start to see these dramatic changes in gene expression, really indicating that retinoic acid may be very important for this neurogenin 2, islet 1, and LHX3 to drive the expression of motor neuron genes. So just to show you this in another plot, this is basically a principal component analysis plot of these different data sets. So down here on the plot shows the embryonic stem cells where we've induced the expression of islet 1 and LHX3, or all 3 transcription factors together. And you can see they all cluster really close to wild-type embryonic stem cells. In other words, these aren't changing that much when we induce them 48 hours after induction. But if we add retinoic acid on top, we start to see the cells, they start to move towards the transcriptomes of wild-type motor neurons. So now they're starting to move towards the motor neuron lineage. So again, it shows that retinoic acid seems to be really essential for this process. Now, I do want to point out that this is not a direct effect of retinoic acid, because if we just add retinoic acid to embryonic stem cells, we do not turn on all these motor neuron genes. So here I'm just showing you an example of some of the motor neuron genes that we've looked at, and you can see here that you get very minor changes in their expression if we just add islet 1 and LHX3 or NIL. But if we add NIL plus retinoic acid, it really boosts the expression of these genes. But again, we don't see these being induced by retinoic acid alone. So what we hope to test is this model that retinoic acid really has 2 different kinds of functions. The first is this classical posteriorizing function, in which retinoic acid will turn on genes like Hox genes by functioning like nuclear receptors do, where they bind to their nuclear receptor target site and activate genes. But we also think that retinoic acid receptor is functioning as a transactivator for EBOX binding proteins. And in this case, we show a model of RAR bound to retinoic acid transactivating neurogenin 2 and E47. So we expect to see that when we chip retinoic acid receptor under these conditions, we should detect both types of binding. So these studies are actually currently happening in the lab. So the next set of experiments, we really wanted to address what happens when we turn on these transcription factors in different cell types. So what we did is we take our inducible mouse line and we cross it with a reporter line that will allow us to determine whether or not we're turning on a target gene of these proteins. And the idea is then we can add doxycycline at different times during development to see what happens when we turn on these transgenes in different developmental stages. So something quite striking happens. If you inject the cells quite early, so in other words, if we inject the embryos at E8.5, you basically see a complete failure of embryo development, and virtually the entire embryo turns green. This suggests that these 3 transcription factors can basically convert almost any cell type into like a motor neuron-like cell. They turn on the transgene in response to NIL. But as development progresses, and if we wait to add doxycycline, you see the phenotypes get progressively less severe. So if you're injecting them at E9.5, you still see problems with embryo development, but the GFP is not everywhere. And again, if you inject it at E10.5, you can again see the embryos are they're roughly the right size, and you do see some expanded GFP, but again, it's not completely pervasive throughout the actual embryo, suggesting that some cell types are responsive to these transcription factors while other cell types are not. So to begin to look at this a little bit more closely, we can do cross-section. So this is a wild-type mouse. And we can see normally that HB9 is only expressed in the ventral lateral horns of the neural tube. But in these INIL mice, what you see, where you turn on this islet-1 LHX3 transgene throughout the embryo, you see that some parts of the tissues are actually responding. So for example, you see in the neural tube, you see dramatic induction of HB9 all the way throughout the neural tube. But you also see some peripheral tissues turning on the HB9 transgene. And we wonder whether or not some of these tissues that might be turning on the transgene, we're getting them in these places because these are areas where we have high levels of retinoic acid signaling. So it's possible that retinoic acid is essential for the induction of target genes by the NIL cassette. So how are we going to address how these 3 transcription factors associate with DNA in different cell types? Well, basically what we're doing now is we're trying to derive many different types of cells from these inducible mice. So for example, we derived embryonic stem cells, we derived primary mouse embryonic fibroblasts, as well as tail tip fibroblasts. And we can show that the system works really beautifully. If we add doxycycline, we're able to turn on our transgene in virtually any cell type. But what you'll see is that these different cell types respond quite differently to the transgenes. So in embryonic stem cells, we can get rapid conversion to an HB9 positive cell, whereas in embryonic fibroblasts and tail tip fibroblasts, we basically see no reprogramming. In other words, the chromatin state is in such a way that these cells are not responding to the transcription factors that we're inducing. Now, we hope to expand this to many other different types of cells. So, for example, we may be able to convert a motor neuron, a neural stem cell from an adult tissue, which has a more related lineage, perhaps could be turned into a motor neuron-like cell. But I will point out that we also know that some cell types in early development seem to have some interesting responses to the NIL transgene that we might not have expected. So for example, if we did our injections at E9.5 and we take the fetal liver, which contains the hematopoietic stem cells, we basically completely lose the hematopoietic stem cells if we induce the NIL cassette. So normally, hematopoietic stem cells have high levels of c-KIT, and if we isolate the fetal liver cells after induction of the NIL cassette, we completely lose these fetal stem cells, suggesting that our transgenes are having some effect on the hematopoietic stem cell compartment. So what are we going to do next? Well, what we're trying to do now is to map the genome-wide binding profiles of NIL and all of these different cell types and try to integrate this data with epigenetic marks to see what might be predictive of whether or not a cell can respond to a certain transcription factor. So in the last slide, I'm just going to tell you that the other interesting thing that we can do with these mice is we think that we can perhaps build better models of motor neuron diseases. So what we've done is we've crossed our INIL mice that have this MNX1 GFP reporter with SMA mice. So SMA mice, these are mice that have mutations in the SMN1 gene that allow us to model a motor neuron disease, spinal muscular atrophy. So the idea here is we can derive embryonic stem cell lines, and within about a week, we can generate a nearly pure population of motor neurons that are carrying genetic mutations that will allow us to model, allow us to do biochemistry to look at the causes of motor neuron disease. And just to show you that the system works really nicely, if we just use patterning factors to differentiate embryonic stem cells, we'll typically get about 30% of cells becoming motor neurons. But if we also include our induced transcription factors, we get a nearly pure population of motor neurons. So this basically tells us that within about a week, we've dramatically improved our ability to create the desired cell types. So just to summarize, induced transcription factors can have strong synergistic effects in embryonic stem cells. They have different outputs in different cell types. we can use inducible factors as a model to study the influence of the epigenome on transcription factor occupancy, and potentially transcription factors can be used to build better in vitro models of disease. So just to thank those who contributed to the work, Carson Miller is the postdoc who did most of these studies, and Wes Gifford and Sam Pfapp's lab helped to get this project underway. He designed the inducible cassette with me, so I'm happy to take questions.
Two quick questions. Go ahead, shout.
Okay, in terms of your model
Be easy with the microphone.
excellent research.
Ron, be easy with the microphone.
Okay, in terms of what you've done is where you've been able to really show as a embryonic stem cell goes through a certain lineage to a ectoderm, and you've isolated each particular stage in changes in transcriptional factors, but you haven't actually said what are the actual genes that are being turned on and off as it goes through that particular stage, and what are some of the microarray changes that are associated with that. Now, have you done that research, or do you plan to do that research? Is anyone doing that research?
Yeah, so we're basically profiling all the cells under the different conditions. So I showed you some of the RNA sequencing plots. So it's clear that the cells aren't just becoming motor neuron-like. In many cases, they're becoming motor neurons. They're indistinguishable from an RNA transcriptome profile from motor neurons isolated from embryos. That's when we do the experiment with ES cells. If we do them with fibroblasts, like I said, nothing really happens to the cells. And we're trying to understand why is it that a fibroblast is completely not plastic? Why can't it be converted with these transcription factors? Whereas if you turn on MyoD in a fibroblast, MyoD can transform them. So we think that there's a chromatin barrier, and that's what we're trying to understand.
A real fast question. Real fast answer.
My question is that using transcription factor reprogramming is not like biological reprogramming, is different.
I'm sorry, I can't catch. Say that more
I
slowly.
just said transcription factor reprogramming is different from biological reprogramming. The efficiency is very low, and the most time the big problem is that the reprogramming is not complete. So usually there is a group of gene determining the phenotype. So how do you determine what group of gene control the motor neuron phenotype? And do you get the true motor neuron phenotype or not? You generate different cells.
So I agree. So for the forward programming, in other words, taking an embryonic stem cell and forcing it to a motor neuron, using these 3 transcription factors, the motor neurons that we get are capable of being re-injected back into a spinal cord and innervating target muscle. So from a transcriptome perspective, you can basically not tell the difference between them and real embryo-derived motor neurons. But I agree with you in that if you're trying to convert a fibroblast cell, we cannot do it yet. And the point is that there's some restriction to this type of reprogramming. So I think for whatever cell type you want to generate, what we're trying to say is that we would like to be able to predict which transcription factors you could use to be able to generate your desired cell type. Obviously, we're at the very early stages. We picked a model system, the motor neuron model, where we already thought we knew what the transcription factors were, so it makes it a nice model. But there are many, many other neural subtypes that we don't yet know which transcription factors would be useful. So we want to improve from the sort of trial and error to being able to have this, think of it like an engineer. Can we just predict and do it in one shot without having to go through 80 transcription factors?
Sam?
All right.